In this video, we're going to talk about enzymes. So we're going to start out by talking about what an enzyme is. So enzymes are typically made up of proteins. So remember, proteins are one of our biological macromolecules. So not all proteins are enzymes, but most enzymes are proteins, although there are some exceptions to that rule. Enzymes are also called catalysts, and a catalyst is something that speeds up a chemical reaction. So an enzyme is one type of catalyst. There are other catalysts out there, but the type of catalyst we're focusing on are the enzymes. And the nice thing about enzyme names is that they kind of tell you what they do. First of all, most enzyme names end with the letters ASE. So for example, we see amylase, lactase, lipase, protease, DNA polymerase. But as you can see here, there are some exceptions to that rule. The other part of the enzyme name also kind of tells you what that enzyme works on. So for example, let's look at protease. So protease is an enzyme that breaks down proteins. So we could write that like this, a protein being broken down into its amino acids. And protease is the enzyme that helps to speed up that chemical reaction. Lipase is an enzyme that breaks down lipids into their smaller components. Lactase breaks down a sugar known as lactose. And people that are lactose intolerant do not make that enzyme. And since they don't make that enzyme, they're not able to break down lactose. And lactose is a sugar that's found in dairy products. And so people that are lactose intolerant, if they eat or drink something with dairy in it, their bodies can't break it down and it causes some gastrointestinal side effects. Amylase breaks down something called amylose, which we also call starch. And then DNA polymerase, we're going to take a look at this one a little bit later when we talk about how DNA is made. So DNA polymerase is the enzyme that builds molecules of DNA. So it helps to build that double helix of DNA. Now, whenever we write a chemical reaction out, the substrate, which is also known as the reactant in some cases, is always written on what I like to call the butt end of the arrow. So not the pointy end, but the end of the arrow that doesn't point. And then there's a line drawn that points to the products that are produced. And then the enzyme is always written above the arrow. So whether it is a substrate getting broken down into products or multiple substrates being bonded together to form a product, the enzyme always gets written over the arrow. So for example, on one of the previous slides, I showed how we can take a protein and break it down into its amino acids. And then over the arrow, we would write the name of the enzyme, which is a protease. So to kind of make that maybe a little bit clearer, the substrate in this case would be the protein. The products would be amino acids. And the enzyme would be the protease. So you might be wondering why chemical reactions need to be sped up. Because chemical reactions happen spontaneously, but that doesn't mean that it happens quickly. And there's actually a little bit of energy that has to go into a chemical reaction in order to break a bond. And so you can see this here in this area known as the activation energy. And so this activation energy is the energy that goes in to kind of help to destabilize the covalent bonds. So these covalent bonds are strong bonds, and so there has to be a little bit of tension kind of applied to them to help them break. So it's kind of like with trying to break a toothpick. It's easier to break it if you kind of bend it in half and kind of put some tension on the toothpick. So what enzymes do is they take the reactant, which remember we can also call that the substrate, and it helps to put a little bit of stress on those covalent bonds to make it a little bit easier for the reaction to happen. So instead of having to have this much energy in order for the bond to break and form the products, instead it might only take that much energy. And with this smaller amount of energy, it means the reaction is more likely to happen in a quicker time period. So most reactions would take too long in order for, or it would take too long to be useful for life. All right, so our day-to-day life, we're dependent on these chemical reactions. We have to make stuff, we have to break down stuff, we have to process energy. And to do all that, we need to be doing these chemical reactions pretty quickly. So we need enzymes to help speed up those chemical reactions so that they happen fast enough to keep us alive. This slide shows you that reduction in activation energy that enzymes do. So you can see how the amount of activation energy in an uncatalyzed reaction is a lot more than the amount of activation energy in a catalyzed reaction. So the enzymes just make these reactions happen faster by decreasing how much energy has to go into the process in order to get to the output of the product. So now that we know what enzymes do, now we need to take a look at kind of how they work. So enzymes are proteins, and if you remember, the tertiary structure of a protein gives it its 3D structure. And it might be the tertiary structure, or it might be the quaternary structure. And this 3D structure matters. Enzymes and their substrates fit together like a lock and key. So you can see on our example over here, you can see how the enzyme has this area here. Let me switch colors real quick. It's got this area here that matches the circular substrate. So it matches this one and this one. Whereas this one over here has this active site or this binding spot that matches these other substrates here. So if one of these substrates, so let's look at this blue rectangle here, if it tried to bond with this enzyme, it wouldn't work because it doesn't fit. But it fits perfectly with the active site over here. All right, so enzymes and their substrates fit together like a lock and key. Or sometimes they call that you might see it referred to as puzzle pieces. So they fit together very specifically due to their 3D shape. So like I said, that 3D shape is really important. And there's one particular spot on the enzyme called the active site, and that's where the enzyme interacts with the substrate. So that's this area here that I kind of outlined. I got a little ahead of myself. So what happens is that when these substrates bind to the enzymes help to orient them in a way that makes it easier for bonds to form or bonds to break. And so I like to think of enzymes as either matchmakers or homewreckers. So a matchmaker is something that's going to make a bond. So if you think about it, let's say that we've got these two individuals here. So we've got this red circular substrate and we've got this green circular substrate here. Now, matchmakers back in the day before we had online dating sites like Match.com and eHarmony, there were actually people that lived in a village that would help to make matches between young single individuals. And oftentimes they would take into consideration things like their family backgrounds and ancestry and stuff. But their goal was to help to bring the two people together and form a match. So to make a couple and they get married and have babies. But I mean, nowadays we have things like Match.com that kind of do that for us. Now, enzymes are matchmakers in that they bring these two substrates together. And in bringing them together, it makes them easier to form those bonds with each other. Now, the interesting thing to take note here, though, is that the enzyme does not become part of the product. And that's one reason why we write the name of the enzyme above the arrow. And that's because it does not become part of the product. So just like a matchmaker brings a couple together, but then the matchmaker is not part of that relationship anymore. The matchmaker goes off and finds two other people to bring close together. So after the enzyme brings them together and allows them to form a bond with each other, so we would end up with a red circle bonded to a green circle. And after that happened, this enzyme here would be able to go off and do that with another one and maybe form another bond between another red circular substrate and a green circular substrate. All right. Now, the homewrecker part means that some enzymes can be catabolic. Yes, so some enzymes can be catabolic, meaning that they can break those bonds. And so they can actually break up the larger molecule into smaller molecules. And so it can go either way, but it really depends on the enzyme and the substrate. So enzymes that are catabolic are different from enzymes that are anabolic. As we mentioned before, enzymes are specific to the substrates that they can act upon. And that's because the enzymes and the substrates fit together like puzzle pieces, or like that lock and key model that we talked about. And what that means is that an enzyme can only break down a particular type of molecule, or it might only be able to bring together particular types of molecules. For example, amylase can only break down amylose. It doesn't have any effect on lactose or sucrose or glucose or any of those other sugars or lipids or proteins or anything like that. Lactase only breaks down lactose, but it does not have any sort of effect on amylose because it only fits with lactose. Lipases only break down lipids, and the name tells you that, so it starts with LIP, just like lipids. And proteases only break down proteins. And so this is one way that our body really controls which of these chemical reactions are happening. And it's based on how many of these enzymes are present, because our body can make more or fewer enzymes depending on what we need them for and homeostasis, and just based on what kind of substrates are available.